Welcome to Cellular Respiration.
Alright, so in the human body, we use two main electron carriers. Actually, no. Ignore that. I did not say anything. Easy. Alright. So one thing that's going to be very important throughout this presentation is the concept of electron carriers. So each electron, I have this handy visual aid, is full of energy. Just look at it. This is a very energetic boy. But you see, you can't just have energy just floating around and stuff, and especially not ionic energy. That's unsafe. So you gotta bind it and carry it with something else. So we have the two main electron carriers of the human body, NADH and FADH, for which I have provided this handy visual aid. This is NAD+. This is positively charged. It's empty. See? Look at all this blank space that you can just fit something in, like an electron. So, a lot of cellular respiration is, in fact, putting this electron onto this NAD+ to oxidize it to form NADH or FADH, and this is the main function of the Krebs cycle in glycolysis to generate spare electrons for these carriers to transport, to oxidize NAD+. So you can use them in the most energetically favorable step of the cellular respiration, which is the electron transport chain. And yes, this is the penguin, reminding us that nicotinamide is related to some other things.
Glycolysis is a very important process in human and other cells, as it is the stage where ATP is created. Glycolysis is the process when glucose, sugar, is broken down into pyruvic acid and ATP is released. The first 5 steps of glycolysis use energy to convert glucose into G3P. Then, the G3P is converted into F6P and energy is released in the form of ATP.
Hello, this is Zeke, here to educate you on oxidative phosphorylation. Oxidative phosphorylation is the final stage of cellular respiration. It consists of 2 parts, the electron transport chain and chemiosmosis. Briefly, the electron transport chain, or ETC, is the transfer of electrons through a series of protein complexes, from electron donors to electron acceptors. In chemiosmosis, the electrochemical gradient produced by the ETC is harnessed to yield ATP. At this point, I would like to take the time to thank the sponsor of this portion of the video, glycolysis and the citric acid cycle, without which we would not have the NADH and FADH2 necessary in the electron transport chain. Stick around to the end of this section to find out more. The ETC is made up of membrane-embedded protein. The 4 structures you can see here labeled 1 through 5 are the protein complexes. NADH and FADH2 from our sponsors, the citric acid cycle and glycolysis, serve as electron donors. As electrons are donated to these complexes from NADH and FADH2 and move through them, they release energy. That energy is then used as hydrogen ions are pumped from the mitochondrial matrix to the intramembrane space, creating an electrochemical gradient. Finally, the electron is passed to oxygen, which splits into 2 oxygen atoms and accepts protons from the matrix to form water. After NADH and FADH2 pass on the electrons, NADH plus and FADH are recycled back to our sponsors, glycolysis and the citric acid cycle. Chemiosmosis harnesses the flow of hydrogen ions down the electrochemical gradient through the only available channel, ATP synthase, which catalyzes the addition of phosphate to ADP, resulting in ATP. Cellular respiration yields around 30 to 32 ATP, 80% of which are generated by oxidative phosphorylation. Now to our correspondent inside the cell.
occurs in two different forms, alcohol fermentation and lactic acid fermentation. Both are anaerobic processes, and they occur after glycolysis in the cytoplasm. Humans and animals perform lactic acid fermentation, while alcohol fermentation is performed by yeast, alcohol, bread, that sort of thing. The key difference between the two is the presence of ethanol and lactate in, ethanol in alcohol fermentation and lactate in lactic acid fermentation. Specifically, alcohol fermentation is a dead-end reaction, so it's irreversible. It recycles NADH and produces NAD+ with a net of 2 ATP. Lactic acid fermentation is reversible. It is anaerobic as well, as both are. Also has net of 2 ATP, and sort of, as you can see in the slide, like the whole process, and then lactate is converted back into pyruvate as soon as O2 is introduced. And it also recycles NADH and sort of has NAD+ as a product. Animals perform this every day, and it normally will occur like after exercising in your muscle cells. So really, that's about it. They can, that's about it for fermentation.
Wake up! 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 Now! Wake up now! Now! Wake up!
And now, the intro to the citric acid cycle. The next step for the newly created pyruvate is to be transformed to take part in the citric acid cycle. And so for that, I have this handy visual aid. Everybody say, ooh. Everybody say, ah. So, now you have this pyruvate, but, the cycle is very exacting. It needs the right molecules to go into it so you can oxidize all those NAD pluses. So, what it does is it goes through pyruvate dehydrogenase, which is basically a process that puts a CoA group on it to turn it into acetyl-CoA, so it can be used in the citric acid cycle and transmuted into citrate through citrate synthase. So what this enzyme will do is it'll just attach a little group on and wax them off, but our handy visual aid doesn't represent that. And then, voila, there you go, you have an acetyl-CoA. Also. And then, here's the penguin reminding us that Flawless should probably be paying attention. In the citric acid cycle, acetyl-CoA, our visual aid from earlier, is first transformed into citrate, which then goes through a series of reactions which, in their most important function, oxidize several electron carriers, that's our NAD+ and the NADH from earlier, and output carbon dioxide. The main function of this is as it relates to respiration, is to oxidize electron carriers. However, all of these little things that's creating, like QH2, GDP, and CoA, are very useful for different parts of amino acids in the human body. And it does generate a bit of energy, but, not a super amount of it. Makes you think about where that fish is right now. That penguin.
Find.
Alright, Zeke, I'm about to start. You're gonna have to cut this bit out because I gotta, move stuff around. Right,
yeah, it's fine. I can cut it out if it's weird. It's not gonna be hard for me.
Jameson, your mic makes you 3 octaves deeper.
I know. I'm gonna put it. I can also boost it too. Should I boost Jameson a little bit?
I don't know, dude.
I don't know.
It might just be because I talk in a deeper voice.
I actually don't know how to do audio very well. I should have Nick do it.
So it's in. Okay.
Garrett's.
Yeah, this is Ox, right?
It's Nicholas Ian.
I'd swear I read it that way the first time.
Alright, so I'm about to start.